Good afternoon, everyone. My name is Kaiwen. I'm a third-year PhD candidate from the University of Michigan, supervised by Dr. Yuan Guan. And thank you for welcoming me to present our write-ups for the Tag 3 of the REX Data Challenge. So my today's presentation will be split in 4 parts. So after a brief introduction about the data and method we have used, and we will first validate the NetraAI findings, and then to present our new observations from the processed and full data. And at last, we will summarize our primary hypothesis based on our new observations and the previous literature. So the provided data actually includes 2073 records. The number is different from what just Jack introduced, is that because some of the individuals may have multiple records in the data. And this data will be used by the NetraAI developed by the NetraMark, which is an AI company. That the algorithm is used for inferring the causality of the symptoms and the diseases. Actually, the data actually have very limited sizes, and they have a lot of data processing on the data, and they only kept about 400 symptoms in their final data set. And with this data set, they propose some findings related to the immune functions. The first one is that they found that the 41 individuals were characterized by the immune-related symptoms, and then a subpopulation of 4 individuals stood out due to the other features like the heart issues, dental issues, bladder issues, etc. And at last, they claimed that the co-occurrence of the immune issues and the other symptoms. So from the raw data, we just extract some columns that related to the symptoms, which was used by the NetraAI. There are 42 columns we extract from the OSDC data map. And we will use this data for validate and hypothesis. We use both the full data and the processed data. So to validate their claims, we may ask 2 questions. The first one is that can we use the symptoms to unsupervised cluster the individuals? And the second one is that can we observe significant symptom difference among the individuals? So for the first question, we will use the UMAP to unsupervised clustering the individuals. And for the second questions, we will use the proportional z-test to make a statistical test the binary features. So let's get to the 3 claims. All of the claims are just directly quoted from the original statement. The first one is that there are 41 children were characterized by the immune-related symptoms, and our analysis agree with that. First one, we look at the UMAP results, where the red dots is 41 children, and the immune-related symptoms have successfully clustered together and separate them from the other individuals. And when using the z-test, we can find that the 41 individuals have significantly higher frequency than the other individuals. But we cannot agree with their second claims. And actually, before the statistical test, we really doubt if the 4 individuals is valid size to make a statistical inference. And our analysis also shows that using the 2 sets of the symptoms, the UMAP cannot separate the 4 individuals out of the 41, out of the others. And when we dive into the 41 individuals, we can also not visualize. We cannot observe the 4 independent individuals from the other 37. And we may curious about if we just look at the other dots that clustered out of the others to see if the 4 subpopulation will include by the dots, but actually it's not. The reason is that there are only like about 7 to 9 individuals that were clustered out of the other 40 other individuals, and this size is also not big enough to make a significant statistical inference. And the second reason is that when we just look at the eyes, not from the statistical side, we can find that some of the heart dental or bladder issue actually have a contradictory conclusion to their claims. The special individuals of the 9, the special individuals may have a better situation of this symptom. So as of their 3 claims, they claim that the occurrence of the increased immunodeficiency with other distinct symptoms among the identified subset of 41 children. Actually, we make a correlation analysis by summing up all of the shown up in the columns of the symptom and sum them together and calculate the correlations with the immune-related symptoms, and found that all of the correlation score actually are higher than 0.25, which means that they may have some moderate correlations between the immune-related symptoms and the other reported symptoms. And also, when using the z-test, statistical test, we can find that the 41 individuals do have some significantly higher frequency than the other patients in these symptoms. So if you remember the UMAP we made previously, we can find that there are actually two groups of individuals are clustered separate with the other individuals. We further look at these groups and found that there are 144 more individuals can be clustered by the immune functions. And actually, when look at the z-test, we found that the immune-related symptoms can actually fully characterize these individuals. That means the other individuals in this data set do not have the issue of the immune-related, do not have the issue on their immunology. And we can also observe that this 148 individual can further separated by the immune function into 2 groups. And the t-test shows that the group 2 may have more severe immunodeficiency issues than the group 1. And we just and then we searched all of the symptoms of columns of symptoms and found if there's any other symptoms are significantly different between the 2 groups. Actually, there are 4, and most including the heart issue, the impulsivity, and the neck issues. And when calculating the correlations between the symptoms, we can find that the group 2, which may have severe immunodeficiency issue, may have stronger correlations than with the other symptoms. So finally, finally, we just dive into the frequency of each individual's of the disease. And actually, in the whole disease, the most frequent disease is actually the Weidman-Steiner syndrome, and I think that's why it's much better than the others in in task 2. And we start looking at the WSS as a start point, and we found that it is not only the most frequent disease in the data set, and it actually have occur more frequently than in the group 2 than the group 1. And we checked the gene interactions using the gene mania, and found that the WSS-related gene KMT2A have a direct relationship with the FOXP1 syndrome. And from the previous literature, it said that mutation in KMT2A is the key to the immunodeficiency in WSS, and the FOXP1 actually is key related to the heart disease, neuron behavior disorder. So actually, summarizing together, we just make a very primary hypothesis. Is that the first thing is that the difference between the group 2 and group 1 actually may occur by the Whitman-Steiner syndrome, and KMT2A and FOXP1 pathway may explain why we may have the co-occurrence of the co-occurrence between the immunodeficiency and the other related symptoms. And then the third one is that we may hypothesize that one with the WSS or FOXP1 syndrome may have a higher risk to get the other one than the other patients. And then one more thing that I just found that after I submit the hypothesis to the challenge is that when you look at the last 3 rare disease, there's no individuals actually showed in the immunodeficiency individual show with the last 3 symptoms. And when I checked the gene mania, I found that actually the gene SETBP1 actually also have a direct relationship with the FOXP1 and KMT2A. But because the limited size of the data, we cannot make some valid hypothesis about that. And finally, I will acknowledge the DREAM team to raise such meaningful competition. Actually, this is my first time to reach out the rare X data, the rare disease data, and I begin to know what the patients are facing now. And I also like to special thanks to Carmen from the REX and also Dr. Jirachi from the NetraAI, who provide the ideas and the tools in office hour about the tax-free solution. And also my mentor, Dr. Yuanfang Guang, who helped me summarize all the write-ups. Thank you very much, and welcome to the questions.
Any questions? So you'd use Gene Mania to help identify relationships between genes. How was that tool in terms of ease of use and access?
You mean the access of the Gene Mania?
Gene Mania, yeah.
Yeah, you can just type the genemania.org and just type in the data, the gene you want to see, and if you automatically generate the network includes these genes.
Nice work, very cool. So I saw you have a lot of comparison data that is not significant, and which might due to lack of the sample size.
Yeah, maybe the samples. Yeah.
Do you have a function or possibility in the future that to calculate a power size or to do a power analysis to see how many patients we should collect it in order to get a significant value to see if that is really the case?
Yeah, that's a good idea. But actually, the data was provided by the RedX and provided by the patients. So we can actually can definitely do that, but the data collection is not our control.